very lucky to have Dora from the Collège de France presenting part of her PhD work for us. So she's going to be presenting her recently published paper from Scientific Reports, where she was comparing extraction method efficiency for high-throughput palaeoproteomic bone species identification. So Dora, if you want to start.
Yes, thanks a lot. It's very nice to hear an introduction. Yes, so as you heard, I'm Dora, and I'm a PhD at the Collège de France, and I'm really looking forward to your comments on our study for the comparative extraction method for species identification based on proteins. For those who have attended ISHI, I'm sorry, it's the same slides, but I will try to say a few different things. So one of the main issues in archaeology and the archaeological field is that from a lot of excavation sites, we recover high number of skeletal remains, but we are not able to identify them by their morphology. And here you can see some examples. For example, in Fuman Cave in Italy, we were only able to identify 3.1% of the bones recovered, while the highest percentage was in Le Coté in France at 37%. So we try palaeoproteomics to resolve this issue. And one of the most common approaches so far is called zooarchaeology by mass spectrometry, in short zooms, and it uses multi-TOF techniques to identify bone specimens based on their collagen peptide mass fingerprints. But this procedure is manually, and it requires a lot of time and a lot of effort from the person who actually does this identification. So in 2022, Ruther and colleagues suggested a new proteomic approach called species by proteome investigation, in short SPIN, that uses an automatic extraction approach, and then it uses LC-MS technique, and the identification of the specimens is automatically based on the SPIN algorithm, and it uses a library search or a library-free search based on 20 protein genes. So the library search is based on Patrick's database created for the SPIN paper, and it doesn't contain all the taxonomic assignments, but it has a variety of those. But we'll discuss this later. So when comparing zooms and SPIN, we see that SPIN provides higher taxonomic specificity when the database is available, and it also has higher success rates for highly degraded proteins, because the LC-MS is expected to have higher sensitivity. And as we can see in Patrick's paper, I will try to find how the laser works. If someone has,
we can see your pointer, the white one.
Yeah. Okay, perfect. So here you can see that in Patrick's papers there were 6 unidentifiable bone specimens, but with SPIN they were able to identify 5 of them. However, one of them was unidentified with both methods. What can you see now? Okay. Okay. I will. Yes. Okay. Sorry for that. So in this study, we try to find optimal extraction techniques for recovering proteins, informative proteins from degraded bone specimens with high protein coverage, fast and cheap, to identify hominin remains. And for our paper, we selected 12 bone specimens in total, that they were most of them, they were already identified from previous studies. And 6 of them came from the Ranis cave here in Central Eastern Germany. And they are dated at approximately 45,000 years ago. The temperature in that area is low Central European, and the cave consisted mostly from humic loamy silt. The remaining 6 specimens, they came from Grota da Compañera cave. I don't probably pronounce it correctly, but yeah. It comes from the south of Portugal, as you can see here. And it's older than 50,000 years old, in high Mediterranean temperatures, and the environment of the cave was consist mostly from Jurassic and Dolomitic limestones. So you are probably already aware of this principle, but just for the better understanding of this study, I will just describe the main steps of the protein extraction. So first of all, we have the demineralization step, which basically we want to remove all the hydroxyapatite or any inhibitors that are present in our bone, so we can access the proteins. Then we denature our proteins. So this means that we unfold the 4D and 3D structure of the proteins to access the amino acid chain. And then after this chain is released, we use digesting enzymes to cut this chain into smaller peptides that are identifiable by the LC-MS or MALDI-TOF. And the interesting thing is that those enzymes, they cut in specific amino acids, so we can possibly reconstruct the protein sequence. The final step of the extraction method is that we need to acidify our proteins, so to stop the digestion, and then we purify our peptides, so they are ready for the analysis on the LC-MS. And for this study, we used 6 different extraction methods, and we tested them based on the species identification, the proteomic coverage, and the deamidation. We sampled 50 milligrams of each of the 12 bones, and we homogenized it into a fine powder, and then subdivided this homogenized powder into 6 different cubes of 5 milligrams. So for this, from this test, we were not able to generate results from all of those methods that I will explain later on. So you will see that some of them, they were excluded. So the first 2 approaches are very commonly used for zooms. So the first one has an acidic demineralization step with hydrochloric acid, and then denaturation is performed with ammonium bicarbonate. The digestion is with trypsin, and then we acidify the peptides, and then we analyze them on the LC-MS. The second approach, it skips completely the first step of demineralization, and it's exactly the same procedure from the denaturation step. And this is commonly used in well-preserved samples. The next two extraction approaches are EDTA-based. And for this, we can digest both the solubilized proteins and the bone pellet, which means the non-solubilized proteins. And for this, we have, for the third method, we have only used lyc, and for the fourth method, we used a protease mix of trypsin and lyc to optimize the digestion process. Then, based on the literature, we know that EDTA precipitates in acidic environments, and that's why we tested acidic and neutral pH environments for both of these methods. And as expected, the EDTA-lyc acidic environment extraction approach generated no results for our samples. Also, we weren't able to generate any results for the RANIS specimens with the neutral pH EDTA extraction. Then the fifth extraction approach combines both the demineralization step and the denaturation step with EDTA and guanidine hydrochloride. But we know from the literature that trypsin is not active in high concentration of guanidine hydrochloride. So we have included a step that we remove the supernatant and we dissolve our bone pellet in ammonium bicarbonate solution to have the optimal environment for trypsin to digest. And then again, due to EDTA precipitation, we test acidic and neutral pH environments. So for this, the method with the acidic environment generates no results. So it is also excluded from the study. And then the final extraction approach that we tested was SPIN, like the original extraction protocol, that uses acidic demineralization solution with detergent for optimal conditions. And then it has a reduction alkylation step with an NEM and TCP. And it's the digestion is performed in on magnetic beads. So basically we use magnetic beads to capture our proteins, and then on this solution, while the proteins are attached to the magnetic beads, we add the digestion the digestion solution of trypsin and lyc to perform the overnight digestion. And then again, we follow the acidification and the purification as usual. So for the species identification, we use the SPIN algorithm that, as previously mentioned, it used the in-house SPIN database that contains a limited number of species, and the broader database that it's commonly available. Then we get our MS spectra, and we compare the peptides of our spectra and the database, both of them, and we see where they match or where they don't. And then based on the highest score, we assign them to species. So the side counts is basically the peptides on peptide sequences that they are species specific. And based on this score, we have our win. So for this study, we tried two different max quant searches. The first one was with triptych, as a triptych search, and the other one was a semi-triptych max quant search. And we see that the triptych search has slightly better results than the same triptych, but yeah, it's not significantly different. And but for this talk, I will show you the triptych max quant results. So when we compare the MS2 scans for the identified peptides, we see that like the well-preserved specimens have no significant difference. And with the dashed line here, you can see the spin. We set the spin. Sorry. Yes. So we have the dashed line represent the spin results, and we compared all the other methods based on the spin results. So we can see that for the Grota-Guattari specimens, that they are the highly degraded samples, we weren't very successful, apart from the acidic approach that we were able to have a higher percentage of MS2 identified scans for 4 out of 6 specimens. Then, even though the acid extraction and the ambic extraction are mostly focused on collagenous proteins and collagenous peptides, we see that for one of our samples, with the acidic approach, we were able to identify non-collagenous proteins that we were lucky enough not to be contaminants for the highly degraded specimens. And then for the species-specific site counts, we can also see the same pattern. So we see that with the acid approach, we have higher number of site counts for the highly degraded specimens. I don't talk too much about the well-preserved specimens because they show no significant differences at this stage. So yes, for the species identification, as you can see, for the well-preserved specimens, we were able to retrieve correct taxonomic assignments for all of Rani specimens, apart from the from the EDTA lysine neutral approach. And we also excluded one of the specimens because the taxonomic assignment was not included in the reference database. And then for 3 of the Croton and Compañera specimens, we had no previously taxonomic assignments, so they are also excluded. But you can see that here again, with the acidic approach, we were able to identify 2 highly degraded specimens correctly and 1 wrongly. But we were completely unsuccessful with the rest of the methods. And to test the authenticity of our results, we calculated the deamidation, and so the hypothesis would be that the amidation should show no differences between the 6 methods, but that's not the case. So we can conclude that the amidation, the amidation is effective from the extraction method that we use, and also usually the glutamine residues deamidate at a slower rate than the asparagine residues, but this is not the case with spin. So as you can see here, it's the reverse pattern, and we think that it's most likely due to the reduction alkylation step that is unique on spin. And to conclude, we found out that preservation conditions affect the efficiency of the extraction protocol, and that we were able to retrieve a higher proteome coverage with the acidic approach for the degraded bone specimens. And this shows that standard zooms extraction methods can also be used for the spin analysis of highly degraded proteomes, and that the extraction approach has an impact on amino acid deamidation. So we would advise people who would like to perform extraction on proteomic extraction on bone specimens to probably have a pilot study with a subset of samples, try different techniques, different extraction proteomic techniques for their sample set, and see which is the best. And yes, I'd like to thank all the co-authors, the people who invited me for this talk, the institutions who funded everything, and all of you for your attention.
Thank you, Dora. That was great. That's super interesting and a really, really useful study for everyone, I'm sure. Is there any questions? If anybody has anything they want to ask straight away, you can either put your hand up or you can unmute yourself and ask your questions for Dora. I can also post a link for the paper in the chat as well.
I can start if nobody else wants to. And since I'm a co-author there, I'm not going to ask you about the study. I'm going to ask you some more mean questions. So now that you've worked with a few different methods, what is your opinion on the bead-based cleanup and bead-based processing that is in spin? Because I've heard a lot of different opinions on these extraction approaches that use magnetic beads. And what do you think? What's your gut feeling?
So, do you want me to reply from the archaeology perspective or from the lab user perspective? I
can do both if you want to.
I have 2 different answers for that. So, see, with the protein aggregation capture, everything is automated. So basically, you leave the plate on the robot and it does everything, and then you go half an hour later, you pick up your plate, you add the digestion enzyme, and then you leave it overnight to digest. So from the user perspective, I say go for the pack. From the archaeological perspective, I would say if you have very low preserved samples, don't try that. Or try a method that there are less steps that you can lose, and some that you can lose proteins. Because I mean, of course, we try to elute the highest amount, but the more steps you have on filtering, or the more steps you introduce into the protocol, the more endogenous thing you will lose. It's like either in proteins or DNA or lipids, I guess. So I mean, if you have runs, just go for the pack. It will save you way too much time and energy. If you have something like compañera, then try the manual method, I would say. Any more questions?
Oh,
Yeah.
Pauline?
Yeah, hello. You can hear me?
Yes.
Yeah. My question is that if I understood correctly, the end of your presentation, it means that eventually, if we do for Zooms the acid extraction on like hundreds of samples, and then we want to sequence the whole proteome of specific samples, we can reuse the acidic solution, like the eluted solution we have for LCMS analysis. So we don't have to resample the bone again. We can just reuse the same extraction, right?
I mean, if you purify your samples on a zip tip or on a stage tip, then the eluate, like the supernatant, will be on the tube, and then you can load you can load it on the Evo tip. So you will only use, I mean, depending on the quality of your samples, you will only use 10%. So yes, basically you can redo the LCMSMS as many times as you have the digestion solution.
Okay, thank you.
So you have a question in the chat from Frankie. So she said, how much weight should we give the differences in the deamidation when choosing a method, in your opinion?
I think it depends on the question that you want to address. So if you want to focus more on the modifications of the proteins, then you should definitely take into consideration any chemical modifications that you will introduce into your samples. But if you just want to test the authenticity, then I don't think the extraction method matters.
Thanks, Frankie. I actually do have a question myself as well. So you said, I think you said you use 5 milligrams of powder for all of your extraction methods. Do you think that using powder rather than sort of chunks or pieces, or do you think using a larger sample weight or a different sample weight would have an effect on your results in terms of which extraction protocol you're using?
I mean, I've used chunks, like bone chunks for another project, and I don't really weight them. So I don't test the input, and it doesn't really affect. I mean, I got crappy samples in. So I think I mean, if you can use more, then go for more. If you don't, like if you can use like 10 milligrams, go for that. But I don't think it's a necessity.
Do you think, I mean, this is for you as well, but also everyone else. And so in terms of sample weight, so if you're using 5 milligrams or 10 milligrams, 20 or 50, do you actually, so how much difference does it make if you have 10 milligrams versus 20 milligrams? Do you think that you generally seem to see a difference in your results and whether you get a species identification or not?
Oh, that's hard to say. because it's based on the preservation of the sample. So yeah, I mean, the ideal solution would be like have several different milligrams, different weights, and then do protein concentration measurement before you run the LC. So when you run the LC, you can load the exact amount of peptides to each of the evotips, and then at the end you will have, I mean, you would expect to have the same resolution for all of your spectra. But yeah, I mean, I haven't tested different. No, actually. But it was for Alice, so everything worked out fine.
I can have an answer from a bit different perspective. I haven't tested this on bones yet, but for my PhD, we tested the difference for dental calculus between 2 and 10 milligrams of material, and we actually didn't see a difference. That could be for two reasons. Either there was something in our protocol that was limiting protein recovery, which is very possible, we were using fast, or it could be that dental calculus is just so rich that already in 2 milligrams, you're basically saturated, but you can't catch. But yeah, it would be interesting for someone to also test this for bones with a much smaller and more limited proteome.
Yeah, I was just kind of wondering that in terms of if you know you have a really poorly preserved set of samples from a site with horrible preservation, is it worth taking a larger chunk initially and then it's going to give you better results or not? Because if it's not, then it doesn't matter, right? And you can take a smaller amount as possible. But if you think that actually it might help in giving you results, then is it worth doing from the start? If you know your sample, if your set of that samples from that site is not very good.
I think from this study, the only advice I could give you is that you can load a higher amount of digestion solution on the evotip. So, for example, you can try 50% then instead of 10%, or you can even try the full amount of the digestion. If you don't plan to reuse these samples and you're only interested in getting some results, just go for the 100%.
I'm going to ask you a bit more of a fun question now then. If you were designing a lab protocol from the start, what is the one thing that you would never do that you maybe have tested or seen somewhere? Like, what is the stupidest idea you could do possibly in the lab?
Oh, plenty.
Pick your favorites.
I don't know. From the chemical perspective, and like try to get as much as we can, I would try to avoid changing tubes and pipetting a lot and moving the samples too much. I mean, if we can remove this step, I would be very happy. And one thing that I would definitely do, but it requires some time and it's extra cost, is that I would definitely measure the concentration before loading anything to the evotip.
Have you tried doing that yourself? It's like in terms of the amount of time it takes you to do, like if you're processing hundreds of samples from a site, you think it's still worth doing?
So I haven't tried it myself, but some colleagues did, and it could definitely give you a reason why your samples didn't work on the LCMS-MS. So if you have like 10 samples or 12 samples, I would definitely do that. If you have thousands or hundreds, I would probably try each plate and then have an average. So, I mean, you still have something to work with, but it will limit your time in the lab.
I have another one as well. If you're with the spin pipeline that you're using, so you use the is that right? You use the spin pipeline to for all of the different protocols.
Yes. So
Yes.
we had I mean, I don't mention that on the paper because it was not relevant. So I tried to so we performed full max concerts with all the data, all the methods together, and then I went like method by method and perform another max concerts for each one of them. And then I wanted to compare the results, but at the end there is no significant difference. So yeah, I mean, you can just do everything at once with both database. I mean, spin pipeline is hard to work with, but it's when it works, it's very useful.
Do you I know that there was some when the paper was first published last year and it was a preprint for a while, there's some limitations in terms of the database you're using, right? In terms of the species that are represented. Is that something that is being developed still at the moment, like a more species being added all of the time to the database with the paper, or is this something that people have to manually do themselves to improve it?
There are some groups that they are working on that. I don't know how far they've been, but there are people working on that for sure.
Do you have other questions?
I might have a question. Hi.
Oh,
Hi, everyone. Hi, Dora. Great talk. Thank you so much. And great paper, of course, obviously. So you mentioned at the end of your presentation that you would advise colleagues to test different protocols on a subset of from a collection. Based on your expertise, do you have in mind a set of like a combo set of protocol that you give it a try and that would give you a sort of range of results that would facilitate the choice of the right protocol for your whole assemblage?
yeah, I mean, I would probably pick one of each. Like one, I mean, I would skip the ambic because you already performed that with the acid approach. So one combination would be the acid with the EDTA protease mix and the spin. I think that's the easiest way, and yeah, you can reduce the time in the lab. I mean,
Thanks.
if you have very well preserved samples, you can even skip acidic approach and go directly for the ambic that you will see a day of lab work. But yeah, it depends on your samples.
Yeah. Thanks.
Of course.
If we have any final questions, people can let us know. Otherwise, we are going to skip the first talk in January, in the first week of January, because most people probably won't be back after Christmas and nobody will be in the mood. So we're just going to skip the first week of January, and we're going to have our next talk in the first week of February, when Eleanor Green from the University of York is going to be presenting, and she's going to be presenting a very cool project where she's looking at feces and proteomics from feces compared to ancient DNA from feces. Going to be really cool. So please tune in for that. In the first week of February, we'll be sending a link around. And again, we have a sign-up sheet. So if anybody wants to present on any topic, so whether it's a paper you've published, something you're doing in the lab, a project, something that you think is working, isn't working, absolutely any topic whatsoever, which is relevant for the group, then please sign up and do a presentation. And thank you very much to Dora again for presenting. It was a really interesting talk. And yeah, we'll see you all after Christmas.
Thank you.
Bye.
Thank you very much. Bye. Have a nice evening.
Bye, you too.
You too.